Hi, welcome to XC Square Biomedical and EE Seminar Series. My name is Jenny. I'm an associate professor at School of Electrical Engineering at KAIST. Today, I'd like to introduce one of the topics we work in our lab, which is ultrasound brain stimulation. So many of you have probably heard about electrode arrays or devices that read and monitor brain signals such as EEG, but probably not much about brain stimulation technology. Stimulating brain is as important as reading the brain. We'll briefly look at why we need to develop a good stimulation technology. Brain composed of millions of neurons control our movements, behavior, emotion, and cognition and memory, and is one of the most important organs in our body. However, despite the great advancement in medicine, many aspects of our brain still remains in dark. Because of these unknowns, we anticipate that many technological breakthroughs will come from neuroscience and brain engineering. With President Obama starting the BRAIN Initiative in 2013, many researchers and engineers, not just neuroscientists, are working closely together to unlock the mystery of 3 pounds of matter that sits between our ears. However, it's just not scientific curiosity that's driving brain research. As our society ages and our life expectancy is ever increasing, degenerative brain disorders is becoming more prevalent and has begun to affect our lives and those we care. The first place a brain stimulation technique can be used is in discovering new neuronal circuits. One great example is optogenetics, which is using light to activate neurons with a cell type specific resolution. This technique, which was first used to demonstrate the technique itself by modulating known circuits, is now widely used to activate inputs of neural circuits to discover new circuits and to study various roles of brain regions. If neuroscience continues to develop and more neural circuits are discovered for different brain disorders, we can use these neural circuits to modulate our brain. For example, as shown in the photo here, we can use stimulation tool to activate or deactivate neural circuits involved with a disease of interest. Besides the investigation of neural circuits and development of therapeutics for brain disorder, another interesting application of brain stimulation is brain-machine interface. So with this background and motivation behind, our lab aims to develop a brain stimulation technique that helps people with brain disorder. In specific, we use a slightly different modality than other stimulation modalities, which is an ultrasound. So let's take a look at the stimulation technique that are currently used. Optical stimulation based on optogenetics is a very powerful stimulation tool. Since it transfects cells with opsins that react to certain wavelength of light, it offers very high spatial resolution and enables cell type specific stimulation. The first method that was ever applied on human was electrical stimulation. The most accepted technique is deep brain stimulation, or DBS, for patients suffering from Parkinson's disease. The next stimulation method is TMS, transcranial magnetic stimulation. TMS uses magnetic field to induce eddy currents to stimulate the brain. And because it doesn't need any surgical procedure and everything is done non-invasively, it is very attractive clinical technique. However, like many engineering systems where trade-off exists, we can see the clear trade-off in the current brain stimulation techniques. If you want to stimulate a very small area with high accuracy, it needs to be invasive. However, if you stimulate non-invasively, then there are many restrictions in terms of how you target and where you target. So among these, let's compare only the non-invasive methods. I have plotted how locally you can target versus how deep you can target. And with advancement in technology, electrical stimulation such as TDC, ES is becoming more local, but it seems still limited in terms of target depth. For TMS, since you can't focus magnetic field, it seems difficult to target the brain with a high spatial resolution. So if you stimulate brain non-invasively, does it mean that it's impossible to target deeper brain structures? Does it mean that it's impossible to target brain with a high spatial resolution? And should we be satisfied with these constraints? So we believe that the targeting deep brain structures with high resolutions will be possible in the future if there's a modality that can be focused, and if it can penetrate our body with a long range. So is there such a modality that satisfies these requirements? The left picture shows simulation results of Simple Life, which is one of the well-established modeling tools for human body. So although there's a skull, ultrasound can penetrate well, and you can see the ultrasound focal spot exists in the deeper brain region. Also, as light can be focused through an optical lens, ultrasound can also be easily focused. So the middle image shows the simulation results of electrical, magnetic, and ultrasound drawn by a Tyler group. While electrical and magnetic simulation is drawn in 4 centimeter scale, where simulation area seems pretty wide, you can see that ultrasound image is drawn in scale of only 7 millimeter, and even so, the stimulation area is extremely small. However, compared to other stimulation modalities, ultrasound is relatively new stimulation modality, and there are many technical challenges still to be overcome, such as uncovering mechanisms, overcoming the skull barrier, and targeting with an accuracy. So here's a short history of ultrasound neuromodulation. In 2010, 2 groups have demonstrated the transcranial activation. What this means is that they were able to induce motor response by delivering ultrasound to brain without removing the skull. So at the first year of starting my own lab, we followed their protocols and were excited to find out that it actually worked. Then the technology has evolved from small animals to primates to human. Then more researchers and engineers have got themselves involved in this field and refined the technology. For example, MR-guided stimulation, brain-to-brain interface, and attempts to uncover the mechanism. In 2018, our group was the first group to demonstrate this technology in freely moving animals by using MEMS technologies, which I will talk about in a minute. What's exciting is that just like the optogenetics, now the ultrasound stimulation technique is used to uncover new circuits. And if in the past it has been used to verify this technique itself, now it's used to do new things like uncovering brain circuits and finding therapeutic effects for brain disorders. Our group works on various aspects of ultrasound neuromodulations with the hope that ultrasound neuromodulation technique will be used as a clinical treatment for people with brain disorders in the near future. In specific, as engineers, we try to understand the current technical limitations and try to propose methods to overcome these limitations. The device that generates ultrasound is called transducer. The transducer used in a typical neuroscience lab is one of the simplest form, a curved single transducer as shown here. And for such transducer, the curvature in the front focuses the beam. However, because we use only one transducer to focus the beam, as you can see from the figure, the focal spot is elongated in the axial direction. The lateral direction, the x-axis in the slide, is determined by the operating frequency of the transducer. In the axial direction, you see that the focal spot will be covering more than a single brain region, causing safety and efficacy problems. The problem actually gets very worse when you apply this to a very small mouse brain. The mouse brain is in the order of only 4 millimeter, thus if you use this transducer, it's going to stimulate the entire brain. Although our final goal is to develop a therapeutic means for humans, we're not sure what kind of brain disorder can be treated using ultrasound neuromodulation yet. Thus, it is important that we can apply ultrasound neuromodulation to small animals where they are abundant disease models. Thus, we have proposed a focusing method using multiple transducers. For ultrasound medical imaging applications, we use very fancy 2D array composed of more than 128 transducers. Using fancy beam forming circuits, we can focus the beam and steer the beam freely. However, these ultrasound systems are extremely expensive and now not readily available to regular neuroscience labs. Thus, we propose a method that many researchers can readily put together without large expenditures. If you look at the figure, you can see that by changing the angles of the crossing, the focal size and the volume can change. And among all the ultrasound neuromodulation work on the mouse and rats, you can see that our work shows the lowest axial and lateral resolution. Although this technique seems fairly simple, you can see that there is a clear advantage of this system versus the phased array focusing technique, in that the aperture, the size of the transducer, becomes extremely large in order to achieve the same spatial resolution achieved by our dual transducer technique. To confirm this efficacy of our system, we first observed that crossing two transducers form a circular focal volume by measuring the beam in a water tank, and also extracted a mouse skull and observed the focal spot after the skull transmission. And due to the skull effect, you see a small distortion in the beam area. Also, we have confirmed our system by looking at the success rate of inducing motor response. We observed that compared to a single transducer, by using two transducers, the efficacy of stimulation improved even at the same intensity. In addition, we looked at the direct activation of neurons through CFOS staining. We observed that significantly more neurons were activated at the target region compared to no stimulation cases. The second limitation we wanted to resolve was the fact that current ultrasound neuromodulation system is heavy and bulky. As you can see from the figure, while mouse only weighs around 20 to 30 grams, the single transducer used in the lab is more than 100 grams. So in addition, the space between the transducer and the brain must be filled with a coupling medium. So as you can see from these photos, neuromodulation system is way much larger and heavier than the mouse. Thus, there are many limitations in exploring ultrasound neuromodulation. For example, the mouse needs to be either always fixed in stereotaxic frame or put under anesthesia. With these limited accurate experiments, it's very difficult to see the effect of ultrasound transducer or neuromodulation in treating brain disorders. So if you can observe the neuromodulation effect in freely moving environments, we can uncover the full potential of ultrasound neuromodulation. However, it's simply not possible with the current system. To overcome this limitation, we came up with a very small and light ultrasound neuromodulation system and reported the very first case of freely moving ultrasound neuromodulation in 2018. We have put ultrasound transducer, coupling gel, and wiring all in this very thin plate you see in the photo. And this technology that allowed us to make such a compact and miniaturized system is called MEMS technology. Using MEMS technology, we fabricated capacitor micromachined ultrasound transducer, also known as CMOT, which is composed of a very thin silicone plate which are operated through electrostatic actuation. So CMOT is composed of multiple transducers placed in an array where if you zoom into a single transducer, there are multiple vibrating circular plates all connected in parallel. And each of these circular plates is in the form of a capacitor, where upon application of AC signal, the plate begins to vibrate its natural resonant frequency. As multiple of the circular drum plates vibrate, ultrasound is generated and transmitted to the coupling medium. While CMOT can transmit and also receive ultrasound like a microphone, for our case of neuromodulations, we are using our device only in the transmitting mode. Here's a photo of our CMOT ring array with a diameter of 8 mm that weighs only 0.035 gram. As you increase the applied AC voltage, the beam intensity increases. Looking at the beam profile, you see the ultrasound is naturally focused and formed at the middle of the ring. Here's the video of using our device delivering ultrasound to the motor cortex through the intact skull. After confirming the successful neuromodulation, we also confirmed stimulating efficacy in freely moving case and confirmed that after fixing our device on the skull, mouse freely moved around in the cage pretty well. The third challenge for miniaturization is the collimator. Another large portion of the system is occupied by something called collimator, which plays role of coupling ultrasound from transducer to our body and also allows engineers to adjust the focal distance. And due to this bulky collimator, you can see that it's almost impossible to do behavior studies. Thus, we are currently developing a thin film that can replace this bulky collimator that plays the role of coupling and as well as addition. With our miniaturized system, we are currently also working on to find out the therapeutic effects of ultrasound for sleep disorders. By stimulating the prefrontal cortex with our system, we are seeing interesting changes in sleep patterns and cognitive behavior. Lastly, the final story for brain stimulations, in my view, is a closed-loop system that combines both the brain monitoring and stimulation system. So for example, to treat brain disorders, we can measure the change in brain activities after neuromodulation and analyze these signals to adjust the stimulation protocols. Such automated system can achieve more optimal treatment results. Thus, we are actively working on to develop novel brain monitoring devices as shown here. The photo shown here is from our paper published in 2020. We made a transparent gold-based microelectrode array to reduce the photoelectric artifact. Since our electrode array is transparent with a very low electrochemical impedance without the use of additional materials, we can record brain activity simultaneously with various stimulation modalities. Here's a figure showing the propagation of the brain signals measured using our array, which was initiated through optical stimulation. Today, I've presented research works we do in our lab to develop a new brain stimulation technique based on ultrasound. Ultrasound neuromodulation is non-invasive and still can penetrate deep with high spatial resolution. And being almost ideal in theory, it's a very attractive candidate for the next generation stimulation platform. But being relatively new, there are many hurdles to be overcome and questions to be answered. For example, how do we accurately target the brain when we can't see inside? What's the mechanism behind ultrasound neuromodulation? How do we achieve MR compatibility? And most importantly, does it have any therapeutic effects? So all the work presented in this presentation is not possible without the passion and hard work of our BMM lab students, Korean and international collaborators, and our funding sources. Thank you for your attention.